Welcome, everyone. I've been asked to make an announcement. Will everyone please turn off his or her cell phone and unwrap the candies? Thank you. So welcome, everyone. Welcome to the Simon Center. My name is John Morgan. I'm the director of the Simon Center for Geometry and Physics. Welcome to this edition of the Della Pietra Lectures. As many of you know, the purpose of these lecture series is to bring to Stony Brook and to the Simon Center scientists who are doing innovative, cutting-edge, exciting, important research, and scientists who have the ability to explain their science to a general audience. And we have an exceptional candidate today on both fronts, both counts. Professor Mani Bassler is a professor of microbiology at Princeton University. She's won many awards and has many honors for her work in communication between bacteria, or quorum sensing, as it's now called. Sorry. We do a lot of quantum stuff here. I can't help yourself. So among her awards are a MacArthur Fellowship, the L'Oréal UNESCO Woman in Science Award, the Wiley Prize in Biomedical Sciences, and the Richard Lounsbury Award in Biology given by the National Academy. Also this year, she's a winner of the Shaw Prize in Life Sciences and Medicine. She's been awarded honorary degrees from several colleges and universities. She's a member of the U.S. National Academy of Sciences, and the American Academy of Arts and Sciences, and is a foreign member of the Royal Society in London. She also won the President's Distinguished Teaching Award at Princeton, which bodes well for this evening. And she was one of the nifty 50 scientists and engineers who gave lectures on their science and engineering to high school students and middle school students. So please welcome Professor Mani Bassler.
Thanks, Dr. Morgan, for the generous introduction. Slightly inflated. I am absolutely delighted to be here. I love giving talks in these kinds of venues, and I'm your neighbor down the road, and so I'm just happy to get a chance to come and tell you what my lab is doing a few miles away, and I'm glad to answer questions, about microbiology and also about biology in general. And so I really do appreciate you guys coming out on the first cold night of the year and listening to this story. So I love bacteria, and that will become obvious over the next hour. And what I want to try to do, besides show you that they're remarkable, is to try to convince you that in fact they have tricks that you might not have thought of. And so my main goal for tonight's talk is to try to convince you that bacteria can communicate with each other. If I succeed in that goal, the second goal is to try to convince you that they're multilingual. And the third goal, which is I actually think, which hopefully will be important, is to try to show you that by just studying this phenomenon that I found out about actually on a lark, and thinking about how these bacteria talk to each other, that in fact one can do something very practical. And so where we're going to go in this story is to try to show you that these bacteria communicate with one another, and if you could actually disrupt that communication, you might be able to think about making new kinds of medicines. And so that's the outline of the talk, and I'm going to just jump right in and hopefully accomplish those goals. And so before I do that, I want to just start by making sure that we all understand what bacteria are. They are the most ancient organisms on Earth, so the first life forms. They've been on the Earth about 4 billion years, as far as we understand, and they all look pretty much like this. So this is supposed to be just a cartoon of like an E. coli cell that might live in your gut. And so bacteria are microscopic. You have to look into a microscope to see one. They're too small to be seen with your eyes. But if you look into a microscope, you would see something like that. And they are single-celled organisms, and they're surrounded by a membrane. And that membrane is like their skin. And so it keeps all the goodies on the inside in, and it keeps the outside world out. So it's a lot like your skin. And then inside of the cell is the cytoplasm, which is like some sort of thick goop. And inside of that are all kinds of biological molecules that sort of slosh around, and they keep the organism alive, much like what happens inside of your cells. So they give the bacteria its traits, and they also let it do whatever functions it can do. And what is different about bacteria than all other organisms is that they don't have a nucleus. They only have one piece of DNA. So you have many chromosomes, bacteria have one. So they have one piece of genetic information, one chromosome, that contains all of their genes, which are the bits of information that when they play out, give an organism its physical form and then all of the functions or traits that it can express. And so bacteria only have a couple thousand genes. So scientists, so they're the most ancient organisms. They look pretty kind of primitive. Their morphology are these single-celled, tiny little organisms, not very much DNA to work with. And so scientists have known about bacteria for almost 500 years. And for 480 of those years, they've always been thought to be these asocial recluses. And what I mean by that is that they have no knowledge of their sister cells. So the way bacteria sort of live in the wild, what scientists had known is if you actually watch them under a microscope, they consume nutrients from the environment, they grow to twice their size, they replicate all of their parts, they cut themselves down the middle, and one cell becomes two, and two becomes four, and so on and so on. And so until about 20 years ago, they were all thought to just act as individuals because they weren't sophisticated enough to be able to have sort of social behaviors or collective behaviors. And what I want to try to show you tonight is that that is far from the truth. What we think is that bacteria were probably the first social creatures and that they invented language, if you will, and it's going to be chemical. And that what this chemical language allows bacteria to do is to count their numbers and to act in groups and to carry out tasks that they could never accomplish if they simply acted as individuals because they're too small. And so that's where I'm going. But before I do that, I want to try to convince you that you should be interested in bacteria and these collective behaviors because they play a major role in your life. So on this slide, what I have is a picture, a cartoon that I made that's supposed to be, this is my idea of a human being. And so this is a person, and what I did was I just did a little calculation to make each of these circles represent some number of the human cells that are in the human body. And we know roughly how many cells there are that it takes to make a human being. It's a few trillion cells. So that's how many human cells you have that give you your form and let you do all the things that you do. So it turns out that starting a couple months after you're born, at any moment in your life, you have 10 times more bacterial cells than human cells in you or on you. So if you do the math, since we're in the Simons Center, you're 10% human. I have to try to think of different jokes. Anyway, you're 10% human. You're 90% bacterial by cells. Okay, but I'm a molecular biologist, and so it's a lot more than about the cells. This is the 21st century. It's all about the DNA. And so I've remade this man out of the A's, T's, G's, and C's. These are the bits that are in the famous double helix of the DNA code, right? And so, of course, the human genome has been sequenced. And so we know how many genes it takes to make a human being. There's about 25,000 genes. So that's how many bits of information to make a human. It turns out that you have 100 times more bacterial genes than human genes in you or on you. So you can pick which metric you like. At best, you're 10% human. The truth is you're really only 1% human. The other 99% of you, in terms of genetic information, comes from bacteria. And these are not just passive riders in and on you. You need these bacteria for life. So it turns out that in these 25,000 genes, you don't have that many genes, so you can't make that many proteins or biomolecules. So it turns out that what these bacteria are contributing is a repository of all kinds of proteins and bits of information that you need to keep you alive. And so what we now get is that there would be absolutely no life on Earth if it weren't for the bacteria. Because every higher organism, every plant, every animal, every insect, every human lives its life in consortia with these little partners that are keeping it alive by providing functions that we don't encode in our own genomes, and also because they make what is called a biofilm, which is like an invisible film all over you and also inside of your gut. And that film protects you from harmful microbes and also from the environment. So in fact, we all need these guys to be alive. And so hopefully, after today, right, you'll think a little bit more differently about what it means to be a human. I think it means to live in this community with these invisible 24/7 partners that are working to keep us alive, starting from just a few months after we're born. So that is your relationship with bacteria. And this idea that bacteria live in consortia with us, that's sort of new news, right? So historically, bacteria have been known for what I would have on the top slide. So we've known for hundreds of years, almost 200 years, that bacteria cause disease. So these are just pictures of different species of bacteria taken under the microscope, and presumably you've heard of all of these diseases. So when you hear about bacteria, you usually hear something bad. And that's because it is very clear that bacteria can bring us onto our knees. They can make you sick, or they can kill you if you're infected with a bacterium that doesn't belong there. So that's sort of what they've been known for. But what I'd like to convince you is that they're starting to get sort of some good press finally, because scientists are now getting that there are these harmful bacteria in the environment, but that in fact most of our lives are interactions with bacteria are beneficial because they're giving us this invisible body armor, but they're also doing all of these tricks that we need to stay alive. And so just for a couple more examples, one that you might know about is that when you eat plant food, so like when you eat a salad, you don't have any of the enzymes to digest vegetable material and get the nutrients from that. So bacteria in your gut provide you those enzymes, right? They also make lots of your micronutrients and your vitamins, and vitamins are required for your proteins to work. We also now know that these bacteria that live in and on us actually work hand in hand with our immune system to keep invading microbes out. So these good bacteria, the immune system can recognize them and they say stay, and they take up all the real estate. And then that, it actually mostly just keeps bad bacteria out because there's nowhere for them to live. And there's more to it than that, but we're only now starting to learn about all these beneficial behaviors that bacteria provide to their hosts. And it's just because you can't see them, nobody knew they were there. And once scientists found they were there, they thought they didn't do anything, but now we get that they play this key role. So hopefully you'll have a slightly different view of bacteria. They're not all bad. They're mostly, at least in developed countries, they're mostly good. But whether you want to think about the good things bacteria do, or you want to think about the bad things bacteria do, I want to come back to the question that is at the heart of my lab, which is how do bacteria get any bang for their buck, right? Whether they're keeping you alive or they're killing you, they are itty bitty little critters. So how can it be that they can be so powerful in the world to either keep us alive or to make us sick? And so I want to try to show you that the key to these kinds of behaviors is that bacteria, again, I've said this, they act in groups, they act as collectives, and when they do things together, they can accomplish things that they couldn't do on their own because of their size. And so that's where we're going to, I'll start to tell you about that now. And so what I want to do is to tell you about how this got discovered, because it's kind of a serendipitous pathway from a sort of anomalous observation to this idea that bacteria can talk with each other. So it all started with this animal, which is called the Hawaiian bobtail squid. So this animal is about this big, full grown, and it lives off the coast of Hawaii, just in sort of shallow, knee-deep water. And so this animal, the squid, is nocturnal. So during the day, it buries itself in the sand and sleeps. But then at night, it has to come out to hunt. And it lives in a beautiful one-to-one symbiosis with a bioluminescent marine bacterium that's named Vibrio fischeri. And so the squid feeds the bacteria, and the bacteria give the squid bioluminescence. And so I want to tell you about that symbiosis because it led to the idea of these bacteria communicating. And first I'll tell you about how this works from the squid's point of view, and then I'll tell you about it from the bacterial point of view. Okay, so in this movie what we've done is we've put this squid in an aquarium, and it looks kind of spastic because we have the lights turned on and there's no sediment in that aquarium. So this squid thinks it's daytime and it's supposed to be asleep. And so it's never supposed to be out in the day because it would get seen by a predator and get eaten. So the way the squid avoids being killed by predators in the daytime hours is by doing this. We've now given the same squid some sediment, and it thinks it's day because the light is on, and so it knows it's supposed to go to sleep in the sand. Predators eat you once, so it is important to be invisible, right? So this guy is thorough. Right, and so now in a second it'll retract its tentacles, right, and it can go to sleep. And if I showed you this aquarium, of course you wouldn't know there's a squid in there. Well, there's actually about half a dozen or 10 of them in there, right? So this is a fantastic anti-predation device during the day. It camouflages itself so it looks like the pebbles, and then it buries itself just a couple millimeters under the surface of the sand, and it sleeps. And so that would be fantastic if it didn't need to eat. And so I've already told you at night it has to come out to hunt. And so now there's a new problem, because I've also told you that the squid just lives in this sort of shallow water. So since it's only in shallow water, on bright nights when there's lots of starlight or moonlight, since it's just in a couple feet of water, that light can penetrate the depth of the water the squid lives in. So if it just came out and was swimming around, right, it would cast a shadow on the sea floor when the starlight or moonlight hit its back. It would cast a shadow, and then predators that were under it could see its shadow, calculate the squid's trajectory, and eat it. So it needs a way to be invisible at night. And this is where the bacterium I told you about, Vibrio fischeri, comes in. So what the squid has evolved is a specialized light organ that houses this bioluminescent bacterium, Vibrio fischeri. So now what we've done is we've turned the squid on its back and dissected it, and I hope what you can see are these two blue glowing orbs. And so the way that this works is that under the mantle or the body of the squid is this light organ. It's like a chamber, and it's filled, jam-packed with this one bacterium, Vibrio fischeri. And so they're in there, they're packed in there like toothpaste. They're so dense. They are bioluminescent. And so what happens is when the squid comes out, when it emerges at night, it has detectors on its back, so it can sense how much starlight or moonlight is hitting its back. And then it opens and closes a shutter. It's just the squid's ink sac under the light organ. And so what it does is it matches how much light hits its back from the moon with how much light comes out the bottom from the bacteria. And so it actually uses the light from the bacteria to counter-illuminate itself by matching it to the starlight or the moonlight. And so that when it swims around, it doesn't make a shadow, and so it's invisible to predators. Right? So now it has this way to be invisible during the day, it's under the sand, and it's also invisible at night because it exploits the bioluminescence that's made by this partner bacterium, Vibrio fischeri. So that is why the squid is willing to feed this bacteria and keep it, is because it extends its lifespan. Okay, so that's how it works from the squid's point of view. And so now let me turn to the bacterial point of view. So what happens is every morning when the squid goes to sleep in the sand, it's got a pump that's attached to its circadian rhythm. So when the sun comes up, the squid goes to sleep and it pumps out like 95% of the bacteria. So now the bacteria, there's just a few of them, 5% of them left. It turns out they turn off the light when they're at 5%. But then they grow during the day, and at night they're really thick again, and they turn the light on only at night when the squid needs it. So the question is, how does the bacterium know when it's alone or dilute and when it's at high cell number, so it only turns the light on at night? And the way that works is by the bacteria communicating with one another. So now I'm going to change to the bacterial perspective. So this is supposed to be a cartoon of my Vibrio fischeri cell. So in the morning, right, the squid is pumped out like most of the bacteria. So even though they're in this little light organ, the world is still really big compared to these tiny bacteria. So in the morning, the bacteria are at low cell density. And so they want to have the program of gene expression going that's good for being alone. And in this case, that means don't make light. The squid doesn't need the light. And so what happens is at low cell density, the bacteria are dark. But what they do make are small molecules that you can think of like hormones, and we call these molecules autoinducers. And those are supposed to be the red triangles. So in the morning, at low cell density, the bacteria have light off, and they make and release these small molecules. And the molecules just diffuse away from the cells. And so that the cells can't sense that the molecules are there because they escape. And that says you're alone, and that means don't make light. But then as the day goes by, as I told you, since the bacteria grow and divide and grow and divide, since all of the bacteria are making a share of this hormone, this autoinducer molecule, the concentration of the molecule outside of the cells increases in proportion to cell number. And when this molecule hits a particular threshold concentration, the bacteria detect it, and in unison, all of them infer by detecting that molecule that they have neighbors around, and in unison, all of the bacteria turn on light together. So I drew them blue. So in fact, these bacteria can tell when they're alone and when they're in a community, and the way they do it is by measuring whether or not this chemical is there. And so this chemical is like a word. It tells the bacteria that I have a neighbor, because no single bacterium can make enough to actually detect its own molecule. Right, so you get how this works now. The bacteria only turn on light at high cell number, and then the squid is doing this. It pumps them out in the morning, so the bacteria are alone, light turns off, they grow, light turns on, and then the squid is just doing this over and over in this sort of daily cycle. And we know that we're right about this because we can just collect the cell-free fluids from around the cells, and if we squirt them on these cells, they'll turn on light. So they don't know there's other cells around. They're simply asking, if you will, whether this molecule is present. And so this was the first example that a bacterium could possibly carry out a collective behavior, which in this case is turning on this beautiful blue bioluminescence. And so we wanted to bring the tools of molecular biology to this, and so to find out how does that actually work. And so by looking at the genes and proteins involved, what we realized or figured out, so this is now is the Vibrio fischeri cell, is that what Vibrio fischeri has is a protein. It's an enzyme that makes that hormone, the autoinducer. And that just diffuses out of the cells, and so it increases in proportion to cell number. But there's a partner protein, which is a receptor, that sits on the bacterial membrane. And so this receptor looks just like the hormone receptors on your cells. And they sit on the cell membrane like receptors do. And what they do is that they have a portion, like the nose, outside of the cell that can detect this molecule, and then the fanny, if you will, of the protein is inside the cell. And so when it detects the molecule locks into a binding site on the receptor. Information comes into the cell to turn on the genes that make the enzymes that make bioluminescence. And so this is a really simple communication circuit. The way it works is the more bacteria there are, the more the molecule there is. The molecule fits literally like a lock and key into its partner receptor, and when that key goes into that lock, information comes into the cell, and the bacteria all change their behavior in unison. And so if you're me, you think that's interesting intrinsically, but the reason that I'm invited here is because it turns out that this is not just some ridiculous anomaly from the ocean. In the past couple of decades, based on this Vibrio fischeri story, we and other scientists started to realize that this is not restricted to just this bacterium. There are now hundreds and hundreds and hundreds of cases of bacteria, different species of bacteria, that have been discovered that all have these communication circuits. So they always have an enzyme that produces a signal molecule. There's always a partner protein that detects that molecule. And then together, these complexes send information into the cell to tell the cells to turn on genes or behaviors that are only useful as collectives, but not when bacteria are acting as individuals. And so now we have a fancy name for this. We're a field, and we call it quorum sensing. So the bacteria vote with these chemical votes, the vote gets counted, and everybody responds to this sort of democratic decision-making process. And what we've also learned, again, it's not just about bioluminescence, including in Vibrio fischeri, right? Typically, 200 to 500 or 600 genes turn on in response to quorum sensing. So it's a big deal when a bacterium changes from acting as individual to acting as part of a group. It's sort of like development, right? And all these genes have to turn on in the right order in order for the bacteria to turn on these community behaviors. So this is called quorum sensing. And so that's how it works. A lot of chemistry has been done. These molecules are not red triangles, they are molecules. And so we and others started to look at what are these molecules that the bacteria communicate with. And the very first one that was identified was the one from this bioluminescent bacterium, Vibrio fischeri, because it's the one we've known about the longest. And so in my cartoons, I have it as a red triangle, but this is the actual Vibrio fischeri molecule or word. And then lots of scientists started purifying these molecules from different quorum sensing bacteria, And so here I just have a smattering of different quorum sensing species of bacteria and the molecule that each one of these bacteria uses to communicate. And I hope you can see right away that the molecules are all related to one another, so they all have this ring in common. But then these side chains, these are just carbons, are a little bit different in every single species of bacteria. And what those tiny differences do is to confer exquisite species specificities to each of these molecules. And so what I mean by that is that there is no crosstalk in these systems. So if we take the Vibrio fischeri molecule and put it on this bacterium, Vibrio harvii does not detect it. And likewise, the Vibrio harvii molecule does not do anything to Vibrio fischeri. So what we understand these receptor autoinducer complexes to do is to confer intra-species communication. These are secret, private conversations that bacteria carry out with their kin that allows them to count their brothers and sisters and then turn on collective behaviors. So that is quorum sensing, and that's how it works. And then we also now know a lot about the different behaviors that are controlled by quorum sensing systems. So I told you in detail about how this works in bioluminescence with this symbiont Vibrio fischeri. I just put a couple of the hundreds of examples here. So I'll tell you about this guy. This is a bacterium called Pseudomonas originosa. It's a common bacteria that lives in the soil. You and I encounter it every single day. It doesn't do anything to you unless you are immune compromised or you have cystic fibrosis. So it's an opportunistic pathogen. So you might know that people who have the disease cystic fibrosis have a genetic mutation so they can't clear their lungs, right? So you and I breathe in all kinds of gunk and we have ways to keep our lungs sterile. People who have cystic fibrosis can't do that. So they always have a polymicrobial infection. What happens is that kids that have CF, typically in their teens and for reasons that are completely mysterious, they become permanently colonized by Pseudomonas aeruginosa and they die of a Pseudomonas infection. So people from with CF don't die of CF. They die of a Pseudomonas aeruginosa infection. And the reason is because Pseudomonas gets into the person that has CF, into their lungs, and then it sits on the surfaces and it makes one of these biofilms that I told you about. That's typically the first step in an infection. It covers itself with this matrix, this goop, that makes it impervious to antibiotics. And so that's controlled by quorum sensing, right? It's a community that makes this film. And then as a group, they all release 40 or 50 toxic proteins and enzymes that chew up the person's lung tissue. So the bacteria are just trying to eat, and this is their mechanism. But the problem is that the damage from all of these exoproducts damages the person's lungs so much that eventually they succumb. Right? So Pseudomonas is right, so it controls virulence and biofilms with quorum sensing. And there are now dozens and dozens of analogous cases of clinically or globally relevant pathogens that all control biofilm formation and the collective production of virulence factors by quorum sensing. And so if you think about it from the bacterium's point of view instead of from the human's point of view, this is a fantastic strategy, right? The last thing a little bacterium wants to do is to dribble out a couple molecules of toxin, right? Because they can't do anything. They can't make any impact. And furthermore, these big exoproducts, those are red flags for your immune system. So your immune system evolved to hunt down invading bacteria and get rid of them. And your immune system partially does that by recognizing these products that bacteria make. And so the better idea from the bacterium's point of view is to wait, to count yourselves with these small molecules, to recognize when you have a quorum, and that if all of you release these factors together, you will overcome the human or the animal immune system. So now we have all kinds of cases that if we make mutants of bacteria that can't talk or they can't hear, so if they don't have quorum sensing, they never release these virulence factors, right? So they only do that as a collective. Right? So there's all kinds of cases of that. I put this one other bacteria named Erwinia here because this is one everybody has encountered. So you know how when your lettuce turns like to that brown soup in your vegetable drawer because you forgot it? That's this bacterium Erwinia. So it's the same idea. It is a plant pest. It's a pathogen of plants. And so what happens is that it needs to make a wound in the plant, right, so that it can colonize and make an infection. And so it uses quorum sensing to collectively release all of these proteins that allow it to make the wound in the plant. But then it also does this totally insidious thing, which is simultaneously to releasing its virulence factors, it releases a whole sort of treasure trove of antibiotics that it has immunity to. So what it does is it makes the wound and then it kills off all the competitors that might want to come in and get the goodies, right? And it keeps the wound for itself and its brothers and sisters, right? And so this list goes on and on and on, but I hope the idea that I really want to try to make here is that when a bacterium gives some component out to the world, it never gets its own product back. The world is too big. And so when they make exoproducts, those are the kinds of things that are controlled by quorum sensing, because it's only when the group does it in synchrony that I get the benefit of your work and you get the benefit of mine. So these are called public goods, right? And you share in the actual making and detect and use of these kinds of products. So that is what we think quorum sensing is about. So now you guys understand quorum sensing, and so once we sort of got that far in our studies, we took a step back and we started to think, this is really great if you're Vibrio fischeri and you live in the light organ of this squid all by yourself, right? It's a pristine relationship, this one-to-one relationship. But most bacteria sort of live in the Wild West. And what I mean by that is they typically live with hundreds or thousands of other species of bacteria. So for example, on your teeth in the morning, there are 600 different, that's a biofilm that you know of. You know how on your teeth you have those sweaters? Oh, it's always nice to have me after the eating part, after a meal, right? So anyway, yeah, I'm typically not the lunch or dinner speaker. Anyway, so on your teeth in the morning, right, you have those little sweaters, right? That's got 600 species of bacteria. That's a biofilm. And you brush them off, and the next day they're back, right? And so most bacteria live like that. They don't get to live like Vibrio fischeri, where it's all by itself. And so this is a picture under the microscope of a biofilm. This is human skin. So this is your or my elbow under the microscope. And what you can see is that the skin is covered with bacteria. That's normal. But the issue for my lab is that every one of these different shapes and sizes, that's a different species of bacteria. So if we were right in the first quorum sensing part of this talk, that these molecules that I showed you are so species specific, how do bacteria count if they're only taking a census of their kin? And so we wondered, how can quorum sensing work in contexts like this, which is actually the normal ecology of most bacteria? And so we wondered if quorum sensing couldn't be more complicated than what we had originally found. And so to explore that question, what my lab decided to do was to work on another bioluminescent marine bacterium. Its name is Vibrio harvii. So it is a very close relative of Vibrio fischeri. It's a bioluminescent marine bacterium, but it lives free living in the ocean. So it's not a symbiont. It lives and it encounters all kinds of other species of bacteria. But we knew it only turned on light at high cell density, and I already showed you there was a molecule involved. So we knew it was going to have quorum sensing, and we wondered, well, what is its quorum sensing circuit, given that it lives in mixes with all kinds of other species? And so the reason that I put this picture up is to try to show you the kind of experiment that we did to answer that question. So what you're looking at, right, this is just a flask of Vibrio harvii. These are some Petri plates. This is ridiculous, graduate students, that if you give them Q-tips and Petri plates, this is what you get. Anyway, and what you can see is that you're actually looking at the light that Vibrio harvii makes. So to take this picture, all I did was turn the lights off in the room, and I took the picture with the light that's made by the cells, right? So we're not doing anything to them. If you go in a dark room, this is what you see. And so what's so fantastic about bioluminescence, right, is that it makes this invisible world of the bacteria visible to us, right? We could just see it. And so the logic that we thought we could do to look at this quorum sensing system was we could just make mutants. So we're mostly geneticists, right? And so we thought we could just randomly mutagenize Vibrio harvii, plate it out on Petri plates, turn the lights off in the room, and look for bacteria that were dark under conditions they should be bright, or bright under conditions they should be dark. And just by doing that, we could find the quorum sensing components, right? So it was a really easy experiment. All you had to do was have some Petri plates and be able to turn on and off the lights, right? And by doing that, we did indeed find the components of the quorum sensing system. So this is supposed to be my Vibrio harvii cell. And so what we found, not surprisingly, was there was an enzyme that made an autoinducer. I've actually already shown you that molecule, an intra-species communication molecule, and then it had its partner receptor. But what was surprising to us is when we knocked out the genes for this circuit, this bacterium still made light at high cell density, and there was another molecule outside. And so we went hunting again, and what we found was that there was a second quorum sensing pathway. So we found genes and proteins for a second enzyme that makes another molecule, so I made it pink this time, and it also had its partner receptor. And then all of that information comes in to tell Vibrio harvii to turn on light when these autoinducers are there, and hundreds of other genes. So remember, it's going to turn on all kinds of genes. We're just measuring bioluminescence. That's the gene we're measuring in our experiment. And so we found the components of this circuit, and then we started to wonder, okay, so why has evolution built this circuit this way? Why are there two systems? And what we thought, the idea that we had, is perhaps if these two molecules encode different pieces of information, then having two systems is better than one. If they encode the same thing, having two systems might not be more beneficial than having one. And so we wondered, what is the purpose or the point of this second system? And so to get at that question, we cloned the gene that encodes this enzyme that makes this new molecule, and we sequenced it. So you know how the human genome is sequenced? So you can sequence genes one by one, especially from bacteria. So we cloned that gene and we named it Lux-S, for Luxor, the god of light, and synthase, for the synthase of the molecule. So we got this gene Lux-S, and then just like all of your gene, a human genome is in the database, there are now thousands of genomes of bacteria, because bacterial genomes are small, in a database. So you can take your pet gene, right, and plug it into a computer, and the computer will tell you, does anybody else have that gene? So we took this gene, Lux-S, that we knew made that signal molecule, we put it in the database, and lo and behold, what we got back was all the sequenced bacteria, right? And so we got this who's who of different bacteria. And so you can pick whoever you like. That's pneumonia, that's ulcers, bubonic plague, cholera, staph, strep, bad breath, gangrene, whatever. We got all these bacteria. Now, these bacteria are not making bioluminescence, right? They're doing pathogenesis, right? And so we thought, well, that's interesting that all of these bacteria that aren't related to Vibrio harvii have a highly conserved Lux-S gene. And we showed that every one of these makes a molecule as well. And so we wondered, well, what is this gene that's in all these genomes? What is it? What does this new molecule do in these other bacteria? So we and lots of other scientists started to study traits that were controlled by this new molecule, which I'll call the system 2 controlled traits. And so this again is just a partial list. This goes in the mailboxes in Princeton. This is Lyme disease, that's the gangrene, E. coli 0157, food poisoning. And what we and others found is that this new molecule seems to control biofilm formation and virulence in all of these important pathogens. And so now we thought, hey, we have two different systems that seem to be feeding into controlling these collective traits. So what is this new molecule? So we purified the molecule from several species of bacteria, and what we found is that this is what I'll call autoinducer 2, because it's the second one we discovered. These are just carbons. It's a 5-carbon molecule decorated with all of these oxygens. And so for you, the point that I want to make is not that that's what the molecule is. The point that I want to make is that every bacterium that we purified it from made the identical molecule. So unlike in that first system where each molecule was slightly different than every other, in this case, all of the bacteria make the exact same molecule. And so our interpretation of that result is that this language is exactly species non-specific. We think this is the bacterial sort of Esperanto, the universal language that bacteria talk across species with. Because they're all making the identical molecule, so I can talk to you and you can talk to me because we have a common language. And so what we think now is that bacteria have to, probably all, that's a big word, not all bacteria have been studied, but bacteria have to be built like this, which is that they're at least bilingual, and there's probably, we have evidence that there's many more molecules involved. And so what I mean by that is that they have to have some way to know self, and then they use this molecule to say other. And so the computation that we have shown that bacteria do is the following. We think that what bacteria do, the sort of first simple thing bacteria try to gauge about their environment, is they ask, if you will, am I alone or am I in a group? So they scan the environment with these receptors. They're like little antennas out there. They scan the environment for molecules. And then what they see or don't, they detect or they don't detect molecules, and that begins to tell them to turn on or off all of these hundreds of genes at the bottom of cascade. So first they say alone or together. And then the much more sophisticated computation that we recently showed that they do is they actually measure the ratio of these molecules. And then they change what behaviors they turn on and off, whether based on whether they see a lot of this one, which means a lot of my kin, or if they see a lot of this molecule, which means a lot of somebody else. Right? So we think then they can distinguish self from other, and that they can tailor these programs of behavior based on whether they and their brothers and sisters are winning or whether the enemy is winning. And so they do different things based on the blends of these molecules that they see in the environment. And so this is basically the way that we're thinking about quorum sensing right now. We think it's a lexicon of molecules, and these molecules are something like dialects or something like that, right, that allow these bacteria to communicate when they're in these different mixed groups and to turn on and off collective behaviors. And so what I want to do now for the final part of the talk, now that you understand quorum sensing, is to try to tell you that it's practical to study glow-in-the-dark bacteria, right? And to try to tell you about our efforts now to interfere with quorum sensing, right? So I have made the case, I hope, that these bacteria, lots of bacteria that you have heard of, need quorum sensing to make biofilms and to express virulence factors. We know if we make mutants in these components, they are avirulent. So could we, the scientists, interfere with that communication circuit in order to make new kinds of medicines? And so the idea is there's sort of two ways you could think about this. The first could be if you wanted to make a narrow spectrum. Oh, so I guess I probably don't need to say this, right? You guys know that we have not had any new antibiotics for the really new antibiotics for the past 30 or 40 years, and we have this global problem of antibiotic-resistant bacteria, right? And so the question is, can you think about it differently? Instead of killing bacteria with antibiotics, can you think about behavior modification as a new kind of medicine? And so that's what my lab would like to try to test. And so there's sort of two ways to do it. The first could be if you wanted a narrow spectrum antimicrobial. So if your doctor knew you had a Pseudomonas infection, you would love to have an anti-quorum sensing molecule that targets only Pseudomonas. And so maybe the way to get that is to try to interfere with this intra-species communication circuit. But often, at least at the beginnings, typically when people are initially sick, your physician doesn't know what you have. And so what you do is you get treated with a broad spectrum antibiotic, right, that actually works against all kinds of bacteria, while your physician tries to narrow down what's making you sick, and then you get narrower and narrower spectrum antibiotics. And so maybe you'd like to actually go after this circuit, which has this generic molecule, and you could make a broad spectrum anti-quorum sensing molecule that would work against lots of different bacteria. Right? So we are trying both of those strategies in a number of different species of bacteria. The logic for how the steps you do are identical, whether you're targeting this system or that one. So I'm going to tell you our newest molecule comes from this system, so I'm going to tell you our newest story. Okay, so we're going to try to interfere with one of these systems. And so the bacterium, right, we cannot do this in Vibrio harvii and Vibrio fischeri. They're not pathogens, so there's no virulence to turn off. And so what we decided to do is to work on this bacterium named Pseudomonas originosa that I have talked about a lot tonight, because this is a notorious pathogen. You know, it does these terrible things to people who have cystic fibrosis, people who have burns, urinary tract infections. And then the other thing that's really bad about okay, that's really bad. What's also bad about Pseudomonas is it's a really sticky bacteria. It loves to make biofilms. So now when you hear about, you know, these problems like in the insurance and the hospital industry, you know, that people go in, you know, to get their tonsils out and they get an infection, a hospital-acquired infection, that's this guy. It is the highest cause of nosocomial infections in this country. And that's because it loves to stick to surfaces. So when you get a breathing tube or a catheter or a stent, there's this big danger now that you're going to get a Pseudomonas infection because it makes these biofilms on stents. Right? So it's a pathogen in its own right, but it's also this terrible cause of hospital-acquired infections. So it has a quorum sensing system. I've already told you that. It's this bacterium that we really care about for all of these reasons. So we thought we would use this as the model to test the idea of whether you can think about anti-quorum sensing molecules as therapeutics. Okay, so I've already told you this now. So Pseudomonas uses these intra-species communication molecules to launch biofilm production, that's step 1 in the infection, sit on the surface, and then the group releases all kinds, 40 or 50 or 60 virulence factors that cause damage in the host. Okay, so Pseudomonas, I already showed you this molecule. So in the intra-species communication molecule, Pseudomonas actually uses 2 of these. It has 2 molecules. So one is this one, which has a 4-carbon tail, and the other one has a 12-carbon tail, right? So they both kind of look alike, but the tails are longer or shorter. So we knew that about Pseudomonas, and so what we did was synthetically, we made a molecule that doesn't exist in nature. We made this molecule in a lab that looks a lot like the native signal, right? So if you look at our inhibitor, right, this left part of it looks an awful lot like the signal molecule. But the right-hand part of the synthetic molecule has this big bump on the end. So it kind of looks like the key, except it has a bump on the end. And so what this does is it jams into the Pseudomonas receptors and it acts like a plug. So the key is sort of in there, the plug, makes sort of a lid, and what that does is it prevents the real molecule from getting in, right? So it's an antagonist. It's a competitive antagonist. It competes for locking into the receptor with this molecule, and our molecule wins. So it sits in there, but it doesn't make signal transduction happen. So it just shuts the receptor down. Okay? And so we made it based on knowing what the real molecules look like. Right? So everybody gets that, right? It kind of looks like it. Okay. So anyway, so now I want to prove to you, I can tell you we made this molecule, but I want to prove to you that all this stuff I'm telling you actually happens. Okay? So we wanted to ask, can you plug up the receptor and stop quorum sensing? So you'll remember I've already told you that sort of the first step in Pseudomonas infection is to make this biofilm, to sit on the surface. So what we did in this slide, this is just a Petri plate, and what we did was we just put a little drop of Pseudomonas right in the middle of the Petri plate. And then if you wait overnight, Pseudomonas swarms out over the surface into this beautiful pattern. This is a biofilm. So it swarms, so it started here, and it swims out over, and it sort of colonizes the plate. So now that's just the normal cells, but if we do that, but instead of just doing that experiment, we put our inhibitor into the Petri plate, this is what happens. So now we put the little drop of Pseudomonas there, and the inhibitor is all around it, and so Pseudomonas can't swarm out and make this biofilm. So step 1 looked pretty good. It looked like there might be merit to this idea that if you actually inhibit quorum sensing, you can shut down biofilm formation. So that's part 1. The second part is this group, this collective production of virulence factors. So we wondered, could our molecule work in that context? So these are just 3 test tubes that we took a picture of. So this is wild-type Pseudomonas at high cell density. Pseudomonas is green. And it's green because it makes this virulence factor that's called piocyanin, so cyan, like green. So it makes this molecule that has a green cut tint to it, and that is the molecule that kills human lung cells. So that is the thing that causes the damage, one of the things that causes terrible damage in cystic fibrosis. So this is just normal Pseudomonas, high cell density, quorum sensing has happened, it's released, it's this piocyanin green virulence factor. If we make a mutant in quorum sensing, so we knock out the gene so the bacteria can't talk or they can't hear, now what you can see is Pseudomonas is colorless. That's because it can't count, and so it never releases this virulence factor. But if we take the wild-type bacterium and we add our inhibitor, what you can see is that the bacterium is almost colorless. And so sure enough, our inhibitor can knock down production of this virulence factor by 90%. So both of these things look pretty good. It looks like we can actually stop biofilms and we can stop virulence factor production, at least in a test tube. So the next thing we wanted to ask is, can you do that in an animal? And so the last experiment that I'll show you is here. We have an animal model for Pseudomonas infection. And so what one does is simply measure the percent animals that are alive over time. And so if we give a mock infection, so no Pseudomonas, of course all the animals live. I mean, they'll just live because they're not infected. If we give a Pseudomonas infection, all of the animals die within a day. If we do that experiment, but we add our inhibitor at the same time that we add the pathogen, what you can see is that now the animals live. It's as if we didn't infect them. So what that shows you is that both on the Petri plate and in the test tube, and then in this model animal, if you shut down quorum sensing, you can actually terminate the infection. And so we're actually really, this is our new result, and we're actually really happy about this result because it makes us think we should actually continue to think about this new way, perhaps, of making antimicrobials. This is not a medicine. It's the lead molecule. I showed you the molecule, right? It has all kinds of features that are not good for being a medicine. For example, it's not particularly potent, so you'd have to take a pill that's like this big, you know, and it would kill you. And so anyway, so what we have to do now is we're teamed up with the medicinal chemists, and so there's a lot known about how you build in with medicinal chemistry, properties into molecules that get them to go where you want them to go, that get them to be soluble, that get them to last a long time. None of that and to get them to be more potent. None of that has been done in our molecule. But now we are absolutely taking that molecule and others and moving them forward into this medicinal chemistry effort. And so where we are right, so that is where we are right now. So I've just told you this. So what our first goal is to make our molecule more medicine-like. Okay, and so we haven't done that yet, but we're working on it. The second thing is, remember I told you Pseudomonas likes to make these biofilms on stents and catheters and breathing tubes. So now that we have these anti-quorum sensing molecules, we've teamed up with engineers because what we'd love to do is to dangle these off of materials like plastics or metals or other materials and make inherently resistant materials. Like we could make biologically inspired materials that actually don't get these biofilms on them. And so we're working hard with the engineers to embed our anti-quorum sensing molecules into all kinds of materials in the hopes that we can make things that are resistant to infection. And then the last thing to say is that I already told you this, is that I work on lots of pathogens and we have both of those circuits, right? And so we're doing this kind of work in other pathogens and especially pathogens of underdeveloped countries. And so most of my work is not in pathogens that are in the first world, but in countries that are underdeveloped, or, you know, because those people actually don't live quite as, those people die of bacterial infections much more than people in this country. And so I spend a lot of time thinking about those kinds of bugs and these kinds of strategies. And so then to sort of do a global summary of what I've told you tonight, I hope what you believe now is that bacteria can talk to each other. So they don't have voices, and so the language that they use is chemical. I hope that what you think is the bacteria are multicellular. So you'll remember, if you go back to the beginning of my talk, these were the first organisms on Earth, right? And they have these collective social behavior. They act as gangs, right? And in groups, right? And so what we believe is that by learning about how they do it, what are the molecules, how do they interpret the molecules, how do they send information into the cells, that we're actually going to learn a lot about how multicellularity evolved on this Earth, because we all came from bacteria, and we think that the principles for multicellularity are in these ancient critters. You asked me this before I even got here, right? Is that I hope what you also believe from my talk is that bacteria can distinguish self from other, right? They have molecules that say me, and they have molecules that say you. And again, we would argue that the cells in your body do that, as do entire organisms. You know, your heart cells and your kidney cells don't get mixed up all day, every day, because different chemicals tell different cells in your body what fates to take on and what jobs to do. We think bacteria invented that, and that extends, something like that extends all the way up through humans. And then the practical part is that you can develop these strategies to stop quorum sensing in harmful bacteria, but then to go back to the very beginning of my talk, remember what the thing that is the coolest thing that microbiologists are learning right now is about this microbiota that lives with you and keeps you healthy. And so maybe the real way forward is to actually study that. It's very understudied relative to what we know about pathogens, just because we didn't even know those bacteria were there 20 years ago. And so people are starting to study these beneficial bacteria, and what would be really neat is if we could stop fighting against the bad ones, but maybe learn enough about these beneficial bacteria that if we made pro-quorum sensing molecules and got the good bacteria to talk even better, remember, they are working to keep harmful bacteria out. And so maybe this part of the story is going to be the real way forward in medicine. But that's only at the beginnings now. And then the final thing to say is that I didn't think any of this up. Once we started to think about this business of sort of manipulating quorum sensing on demand, right, we and lots of other scientists said, you know, if we can think this up, the bacteria had 4 billion years to think this up, right? And the idea that I, with a graduate student, I'm going to make a better molecule than what these guys made over 4 billion years is absurd. And so maybe we should stop doing what I just told you and actually just go out into the wild and see what the bacteria have developed, because strategies that we find in the natural world will have won over billions of years of evolution. So those should be the starting components that we bring into the lab and use to make our medicines or products for industry. And so sure enough, what you can find is if you go out and you look in dirt or you look in seawater, one guy will be trying to talk and the guy next to it eats its molecule and uses it for carbon. And so this guy, can't count, and that guy's getting fat, right? And they eavesdrop on each other, they free ride, they make antagonists that are actually better than the ones I make. They have enzymes that cut the rings off those chains, so they do all of this shenanigans out there, right? And so people are now bringing those into the lab and asking what really works, and then trying to start from there. And so I think that's a really fun and exciting part of where this field is going. And then let me finish by showing you the people who did all of the work that I talked about today. So this is my gang at Princeton, New Jersey, right? It's an absolute privilege to get to work with these really smart, creative people who came to the lab not believing what we used to think about bacteria, and they absolutely made all of the discoveries that I told you about tonight. Somebody's brain and hands in that picture did every experiment I told you about. And I see a lot of young people in the audience, and so I want to finish by saying that if you're interested in science, even if you think it's hard, right, you should go for it. You should try it. Challenging is good. You don't want to be bored. You actually want something hard to work on your whole life. And the other thing that I would say is that it's the greatest life in the world to be a scientist, right? My job is to go into the lab and to work with all these really smart, energetic people, nice, energetic people, and to actually think about things that no one on earth has ever thought about before. And that is awesome. We are adventurers. Right? Our adventure is invisible, but there's plenty of science adventures out there. There's so many things to discover. And so if that sort of excites you, that your job is to change the way the world thinks about people think about the natural world, it's a great job, right? We just play games and we try things and we mostly don't understand what we're doing, but every now and again we get these moments of clarity and then these stories sort of emerge. And so it's really, really fun. I just want to tell the young people, we work in these big groups, in these big labs, and it is quorum sensing. Everybody helps each other and the group gets more than the individual. And so I think it's really fun and you should give it a try, because I think that that the other thing that I would say is that discoveries, at least in this country, discoveries get made by young people, right? So these people all come to my lab for 3 or 4 or 5 years, and then they go start their own lab. And so the actual process of science, right, is done predominantly by people between 20 and 30 years old, right? And so that is not so far from some of the faces. It's pretty far from some of the others, but it's not so far from some of the faces that I see in this audience. And so it's not like scientists are these weird, rarefied, Albert Einstein people, right? These are the people that change the way we think about the world, and to me, they look just like you. And so anyway, I want to thank you for letting me come give this talk. That has also been a privilege and a delight, and I'd be happy to answer questions, I think, if you do that. Yeah. Mm-hmm. Sure. Oh.
Questions. One is, do people in your lab, microphone?
No, you should, I just didn't see your,
Do people in your lab have to protect themselves from Pseudomonas after we heard all this about what it can do?
Yeah, they do not. So so Pseudomonas is an opportunistic pathogen. So if you're healthy and not immune compromised, it doesn't infect you. And so so there are rules and regulations, and we follow them strictly. So we wear lab coats, we wear eyeglasses, we wear gloves, and we sterilize our bench every single night. But Pseudomonas doesn't make an aerosol. So when you're sort of slopping it around, I don't mean that really, with pipettes. In fact, you can't, we carefully chose bacteria that are what are called level 2 pathogens, which is you wear gloves, you wear a lab coat, you wear closed shoes, and you wear safety glasses, and you sterilize your bench. Those are the rules, and autoclave all your waste. And then you will not, if you're doing it like that, you won't get sick. And we purposely, my lab, works on that level of pathogen for exactly that reason, right?
The second question is, a lot of talk these days. Doctors will tell you you should take so-called probiotics. And I gather they have a lot of bacteria in them. Is there any relationship between that kind of thing and what you were talking about?
Yeah, there is. And so I will now give an opinion, but it is a scientific opinion. So probiotics are going to be the future, right? So the idea is you take these pills and you can get good, healthy bacteria like in your gut, like yogurt is loaded with them. What is so I think that will be the future. You'll get a little cocktail that's good for you, right? But right now, there's absolutely no evidence that those bacteria ever make it into your gut. And that's good. Because remember, the ones that you have there, unless you've been on an amazing course of antibiotics and your gut is depleted, that's when you can actually make a dent. So this microbiome that you have, yours, I have mine, is so hardy and so robust, we don't know how to perturb it. But that's good, and that's why you don't get sick all the time, because you eat and gobs of bacteria all day, every day, and all the real estate is taken up, right, by your microbiome. And so I think you should eat yogurt because it's loaded with calcium, right? It tastes good, but I think at least for now, the sort of buying of probiotics, that's not useful use of money, but it will be in the future, right? And remember, this part of it, the sort of bacterial that are beneficial, that part of it is just now we're learning that that's there, and it's awesome how hard it is to change that, which is why we're alive, but also that's the problem for the scientists, because we would love to be able to do that. And I think someday we will be able to. It's not long away, it's a decade away, but it's not now. Yeah, yes.
I noticed on your inhibitor molecule you had a halogen.
Yeah. Why? Why? So that was that bromine, right? Why? Because that was the most potent molecule we want. So we made about 400 or 500 molecules, and we just tested them, and that's our most potent molecule, right? And we were just making different, bumps. We put fluorine there, we put bromine there. That one in the meta position was better than para. And we have a crystal structure. We know why, right? It actually locks the receptor into this really interesting conformation that it can't do signal transduction. But that was just fortuitous. We made hundreds of molecules that looked like, the left side looked like the real one, and we put all kinds of bumps, if you will, on the end, and that one worked the best.
I'm going to do a follow-up.
Uh-huh.
Your inhibitor stops that
interspecies
quorum, like the one where they can all talk to.
Yeah, that inhibitor is for intraspecies, but we have the interspecies too. Yeah, I didn't.
If you have an inhibitor for the other one for good bacteria.
Right. Okay. So I will, if you will permit me, I will of course answer the question I want to answer, not the one you asked. But it actually, in this case, it is the one you asked. Right. So your question is, let's say we went after the pink molecule, the interspecies. Right. So if you could shut them down, you'd shut everybody down. Right? And that's your question. Now I've made this big argument that all these bacteria are so good, you might not want to go screwing around with that. Right? And so you are absolutely right. Our interspecies inhibitors shut everybody down. And so what happens is not that much. And so what I will tell you is that, so traditional antibiotics, every antibiotic anybody in here has taken, right? Those kill bacteria or they slow growth. And they are, penicillin, tetracycline, they're not specific. It's not like they just kill, or like if you get an infection and your physician gives you an antibiotic, it's not like it just targets the bad guy. And so you get a stomach ache lots of times when you take an antibiotic. Women get a yeast infection because all of your most of your microbiome gets decimated, right? And what is so the real question is, how did antibiotics ever work? Now that we know about the microbiome, and what is the magic of the human microbiome is that you have these bacteria which are called persister cells. So when you take an antibiotic, right, you kill lots and lots and lots of the bacteria, say, in your gut. But then you have these cells that are identical to the other bacteria, but they're not touched by the antibiotic. And so after you finish the course of an antibiotic, these persister cells start to regrow and they repopulate. And that's why we had antibiotics for the last 80 years. And so now I'll go back to it's amazing, right, that those exist. And that's why antibiotics didn't kill everybody when they took them a long time ago, right? And so now I'll go back to your question, which is, what about if we could make an interspecies? So if we could do it, we made an interspecies anti-quorum sensing molecule, all the chit-chatting in here would be goofy. They'd be all over the map, right? But they would at least, so we know that, but they would at least still be there. And so we are banking on the 80 years of much more extreme traditional antibiotic treatment and knowing it works to understand that if we make everybody goofy for quorum sensing, if you took the therapy off, right, so you're just going to make the bad guys not launch their attack while the good guys are going, what am I supposed to be doing, right? And then somehow your immune system knows the bad guys from good guys, it would clear, and then if you took the anti-quorum sensing molecule off, everybody would go back to chit-chatting, right? And so it's actually, we are banking on what we know about antibiotic use to be able to make the claims that I'm making. Now we have to prove that, but that's actually not our worst problem. I'll let you try and guess what our worst problem is, but that we're pretty sure is going to work fine because we know about real antibiotics. Mm-hmm. Yeah?
About the robustness of the microbiome, what do you think about your personal scientific opinion about the recently very widespread hypothesis that the endemic of obesity is not due to hypercaloric intake, but actually due to microbiome?
Right. So I hope everybody can hear that since it's the back. So there's all of these tantalizing connections, like whether you're obese, whether you have asthma, whether you have irritable bowel disease, Crohn's disease, right? That there's these microbial components, right? And I think that they are increasingly correct, but there's a causation, there's a part that is unproven so far, which is, so what's known is if you're obese, you have this certain kind of bacteria that actually stores more fat. So the question is, am I obese? And so now that bacterium can colonize me, or did I have that bacterium and I got obese, right? And so for sure there's a connection, but it's this sort of chicken and egg, I think you said, problem. And so we, meaning microbiome people, will be able to sort, it's not sorted out yet, but it will be sorted out. So now what we're doing is taking these longitudinal, remember nobody knew these bacteria were there. So you have these obese people, they have these bacteria, but you don't have samples from them before, like when they were babies, before they were obese. And so now what scientists are doing are taking these longitudinal samples. So now instead of saving your baby's lock of hair, you save the first poop, if I may, right? And then you find out what bacteria are there over time in these different environments, in different health contexts, right? And so those things will get sorted out in the next decade probably, but increasingly, those connections are there. And that goes back to your idea. If we could manipulate these bacteria reliably and safely, that is going to be a huge part of what we're going to call personalized medicine, going forward. It is your genome and your microbiome. What is also known is you remember these bacteria are doing all this biochemistry. So like people, you take different medicines and some people have bad side effects and some people don't, and some people the medicine works. It turns out it's because these bacteria are metabolizing these molecules into different things. And so this idea that we're actually going to learn enough about those bacteria to be able to give you medicines that actually are going to work for you and you're not going to have side effects, that's the dream right now in my field, right? And be able to say, no, you're not going to be obese because we're going to give you this anti-obese bacteria, right? That's the hope, right? But it's just the beginning of this field and it's super exciting. I mean, these bacteria, everybody thought, bacteria, if you were smart, you wouldn't work on bacteria because they all got solved back when they found DNA. They're having this renaissance now, right? Because we get that they do all this stuff, right? Yes, it's a pretty fun time in the field. Yeah.
First of all, thanks for a fascinating talk.
Oh, you silver-tongued devil, you're welcome.
My question's going in a totally different direction. Why does Vibrio cardii invest energy in producing light?
Right. Okay, so, look at the time. Got to go. No, I can't ever get out of the room without that. Yeah, okay, so you get his question, right? This is a free, so you get why Vibrio fischeri makes light. It gets something back, food, right? So why would a free-living bacteria make light? And so the real answer is going to be, I don't know, period. And then I'll give you an answer anyway, but that is the real answer, right? So what we do know is Vibrio harvii is older than Vibrio fischeri. So the idea is, bioluminescent bacteria existed, and then Vibrio fischeri got smart and gets a free ride, right? And there's all kinds of symbiotic things like that. And so one idea, the light is blue. You saw that blue is the light that travels far in the water. So everything in the ocean, so it's estimated that 75% of all the organisms in the ocean either make light or use somebody else's, right? You don't have to go very deep and there's no light. And so making light in the ocean is a big deal. On land, there's like 3 things, fireflies, a glowworm, and some fungus, right? So there's no selection on land, not really to do that. So all the light is blue, because that's what travels far in the water, and so Vibrio harvii does that too. So it's as if it wants to be seen, right? And so for sure the light got selected to be seen ultimately. But one idea that people have is that the actual reaction that makes a photon of light, right, that it actually started as it's just the reaction. What the reaction does is it consumes electrons, which is like, electrons, they're like when you guys eat cranberries and blueberries, right? Because they're antioxidants, right? So that's what the reaction makes a lot of, is like an antioxidant. And so maybe it started that it was just the reaction they needed to do to get rid of all of these harmful electrons. And occasionally you give off a photon of light, and over evolutionary time, if some fish sees you and eats you and excretes you over there or takes you, maybe if you get just the tiniest selective advantage, right? Because somebody comes along and eats you, and that's the good life for Vibrio harvii, and takes you to a place where there's not maybe all that reductant, and excretes you, maybe you keep it, right? I mean, that's kind of a crazy answer. We know everything in the ocean makes light, right? Including these free-living guys. And so one idea is it's what's called the dark reactions of luciferase that actually mattered originally.
Good answer.
Yeah, yeah. I actually thought I was going to solve that. That's actually why I started working on that. I'm like, why is this guy making light? And then I got distracted, and so I actually never quite got there. Yeah.
A couple of times that our symbiosis with bacteria in 2 months.
Yeah.
For what do we do before that?
So you come out sterile, right? You're sterile in the womb, and actually you begin to be inoculated on the way out, right? And so you look a lot like your mom's microbiota at first. There's a lot of transfer here. And so what happens is that you start to get inoculated the instant you come out, right? But it takes a few months to get this stable sort of inoculum that then lasts your whole life. And you get it from your mom and dad, basically, the same way you get your DNA, your human DNA. And so you get it with the people you are swapping spit with, right? So I look like my sisters. Actually, I look like my cat now because, right? But when I was younger, I probably looked like my husband. But anyway, so you get it you look like the people you live with, okay? Because you're touching things, right? But it takes a few months to get this sort of stable, hardy population.
But if they're so important to us, how do we survive?
Yeah, no, you survive. You survive. Well, babies get a lot of colds, right? No, you survive, and then of course, remember, you're not digesting all these foods. You're not eating vegetable matter when you're a baby, right? And then you get it. It just takes a few months for it to develop stably, but it starts on the way out, right? Yes.
Is there any sense in which quorum sensing is the precursor to a nervous system?
So I don't know about that. I mean, I'm brainwashed. I think we came from bacteria. Right, we evolved from bacteria, so in some way, bacteria are the precursor to everything, right? And so for sure, the way these circuits are set up, they look a lot like the way neurons detect information and send it into the cell. I mean, that's electrical, but the actual engineering of the circuit where you need, multiple hits to the detector, that works here too, except that the detection is chemical. Right? But so I'm not being clear. The way the architecture of the circuit, the way information gets encoded in something and then transferred in the cell, it is exactly parallel to the way neurons do it, except that's electrical and this is chemical.
And I was just wondering, you have the notion of an action potential. Okay, can bacteria create anything like an action potential, or a cell wall cannot do that?
So they have an action potential across the cell wall, right? Not that I know of. Yeah, right, exactly. Yeah, but they do use that energy change across the cell wall to transfer molecules in and out, by letting out electrons or.
They don't create anything like a nerve impulse.
Not as far as I know, but Who knew, right? Yeah. Other? Yes.
Do bacteria release only one type of inverse species molecule? Do they release different types depending on whether they want to recognize an enemy or?
Right. So that's a good question. So the molecule I showed you says other, right? Because they all make the identical molecule. So it does not encode who the other is. But in fact, I didn't lie, I omitted, right? We know of 3 at least, so there's more molecules, right? You get the idea. There's at least 3. There's a molecule that says me. I showed you that. There's a molecule that says you are my cousin, but you're not me. And then there's this molecule that says other. That's what we know so far. So the idea that the molecules encode number and also something about how close or distantly you are related to me, that's what we're thinking now. So we know 3. There's a me, there's a molecule that all Vibrios make, but nobody else makes, and then this other. And so the idea, but we're not done, right? I'm gonna have a job when I go home tomorrow. And so the idea that there's a whole sort of set of molecules that say, not just that, but say you're my friend that I team up with, and you're my enemy that I, right? Like the idea that they're like little sort of computer chips, right? That are measuring this complicated blend of molecules. We are still hunting for molecules, right? And I don't think we're done, right? So I think that's sort of the frontier where this field's going. But I think it has to be like that, right? Because there's not enough information in what I showed you to make the kinds of decisions that they're making. Yeah.
Have you found anything different? I don't know a lot about this in relation to something that makes spores like.
Yeah. So spore-forming bacteria have quorum sensing, including C. diff has that second molecule. And a better bacterium that's been studied more is Bacillus subtilis. And so what quorum sensing does in that's just a bacterium that lives in the dirt. It's related to anthrax, Bacillus anthrax, but it's like the nice Bacillus, right? And so that bacterium decides, should I switch between being a spore, which is to be this this sort of like a seed and waiting for the environment to get better, or should I grow vegetatively and eat up all the nutrients? And so it is constantly choosing spore or growth, and quorum sensing controls that. So like when they get too crowded, maybe they decide to take this totally other fate, right? And so quorum sensing controls that decision in lots of these spore-forming bacteria. Yeah. So that's the question, right? So you guys know so the question is viruses have quorum sensing, right? So you guys would know what viruses are, right? So they're not really alive. So viruses are entities that they get into other cells, right? And they hijack your components to make more of themselves and infect the next cell. So they need a host cell to replicate, right? So they don't have all the machinery to make more of themselves, and so they live inside of cells. And so bacteria get infected with viruses, the same as we get infected with animal viruses. And so the idea so far, so there's 2 papers, and these came out this year. The idea is, so viruses don't have very many genes, right? Because they are masters at using your machinery, and so they're really stripped down. So the idea is they don't have enough genes to have their own quorum sensing system, but that's what viruses do. And so there's a beautiful new paper, and I'm very proud because it was a person who came from my lab and started her own lab. That this woman, Sina Svenningsson, found that what viruses she thinks do. So sorry, I get really excited. So what viruses do, right, is they grow inside and then they burst the cell and they go to the next cell. And they have to get in the next cell or else they get dehydrated and it's over, right? So viruses would like to understand, if you will, if there's other cells around. If I'm going to burst my host cell and come out, there better be somebody else for me to get into. So she thinks that what they do is that quorum sensing is measuring the cells, that the viruses are tuned in to the bacterial quorum sensing system, so they know when to burst the cell, so they'll have a maximum chance of getting into the next host, right? So they're actually eavesdroppers, right? And so that's the first hint that there's a connection between quorum sensing, but it's the bacterial quorum sensing system and a virus. Right? So you don't want to burst out if they're at low cell density, because then you're out and there's nobody else to infect, right? So that's Sina's idea.
One last question.
Bad for you? No. So all bacteria aren't. So most bacteria that you encounter, you're mostly not sick. So you have 10 to the 14th bacteria in your gut, they're on your skin, they're in your hair, they're everywhere, and they are beneficial, right? Because they give you all kinds of traits, like helping you digest your food, making vitamins, covering your body so that you don't get bad microbes on you, and so most bacteria that you encounter in your life are good for you.
Thank our speaker
Thanks for
once
having me.
again.